Namaste students. My name is Dr. Neeraj Dhulia, Faculty of Agriculture and Veterinary Science, Jayoti Vidyapeeth, Women's University, Jaipur. So in this lecture, we are going to study about the diagnosis of typhoid. Okay, so the topic belongs with the subject medical biotechnology. So let's start. In the previous lecture, we have discussed that what is the typhoid, what is the causative agent, how to prevent it, what are the treatment, and the general information about that. So in this lecture, we will focus on the diagnosis for the typhoid. So the diagnosis of the typhoid may be of many types, like the antibody detection, antigen antibody detection, and specifically the IgM. So the detection of the Salmonella typhi specific IgM antibodies, it serves as a marker for the infection of the disease, and it can be detected as early as 2 to 3 days after the onset of the symptoms. Then blood culture is also there. So the laboratory diagnosis of the typhoid fever requires the isolation and the identification of the Salmonella enterica serotype typhi. Blood culture is considered as the gold standard for the diagnosis of the typhoid, and it is the best when performed within a week of onset of the symptoms. So the multiple blood cultures with larger sample volumes always yield better results. Okay, then the RT-PCR or we can say the real-time PCR is a molecular test for the diagnosis of the typhoid, and it is ideally performed within the first 5 days of the infection. Okay, so out of the different diagnosis methods, the antigen antibody reaction or the antibody detection or antigen detection using the Vidal test is a very famous technique. Okay, so we will study the Vidal test in detail. What is the Vidal test and who discovered the test, when it was discovered, and how to perform this, and what is the principle of the Vidal test. Okay, so it was discovered by the scientist George Fernand Vidal. Okay, so the name of the Vidal test is on the name of the scientist who discovered the technique, George Fernand Vidal, or you can say the GF Vidal. Okay, so this was discovered in 1896, and this is an indirect agglutination test for the enteric fever or the Salmonella type the fever which is developed by the infection of the Salmonella type. So in cases of the Salmonella infection, it is a demonstration of the presence of the O-soma false positive result. So test results need to be interpreted carefully to account for any history of the enteric fever or typhoid vaccination and the general level of the antibodies in the population of the endemic areas of the world. Okay, so false positive means the disease is not there, but you get the positive result in the test. So what are the different possibilities, what are the different cases where you can get the false positive result? So because this is the test who recognizes the antigens or antibodies in the serum. Okay, specifically the antibodies in the serum. So the antibodies may be there if you are vaccinated. Okay, if you have taken the typhoid vaccine, then definitely the antibodies will be there, and if you perform the Vidal test for in that condition, it will get the positive result. It will give the positive result. Then the second case, if you have already infected, means if in the recent history, if you have got the disease before, okay, or you have the history of the enteric fever or typhoid fever, so the antibodies will be there in your body, and in the Vidal test you will get the positive result. Then some base level of the antibodies or may be there, and that may also cause the false positive result. So we confirm the abundance of the antibodies in the body. So specific titer is considered, that if in this concentration antibodies are there, that will considered as the positive. Okay, and below that concentration, the presence of the antibodies are not considered as the positive result. Okay, so this was there the enteric, if you have the history of the fever, if you have the typhoid vaccination, and the lower level of the antibodies, and the populations in the endemic areas of the world. So in some of the areas in the world, uh, it is very frequent to develop the typhoid disease. Okay, so in that populations, the antibodies are generally found in the persons who are living there. Okay, so as with all serological tests, the rise in the antibodies level needed to perform a diagnosis takes 7 to 14 days, which limits its applicability in the early diagnosis. So other means of diagnosing the Salmonella typhi include the culture of blood, urine, and feces, and these organisms produce the H2S from thiosulfate and can be identified easily on differential media such as the bismuth sulfite agar. So that is another thing. In the Vidal test, the 2-mercaptoethanol is often added to the Vidal test. And what is the function of the 2-mercaptoethanol here? This antigen more easily denatures the sorry, this antigen, not the antigen, this reagent or this agent more easily denatures the IgM class of the antibodies. So if a decrease in the titer is seen after using this agent, it means that the contribution of IgM has been removed, leaving the IgG component. So this differentiation of antibody classes is important, as it allows for the distinction of a recent from the old infection. So in the recent infections, IgM antibodies will be higher, and if the infection is old, then IgG antibodies may be there. Okay, so the Vidal test is positive if the TO antigen titer is more than 1:160. Okay, so this is the specific dilution 1:160, and that will be considered as the positive in an active infection. Or if the TH antigen titer is more than 1:160. So in past infections or in the immunized persons, the TH antigen will be considered at the 1:160 dilution. So a single Vidal test is of little clinical relevance, specifically in endemic areas such as the Indian subcontinent, Africa, and the Southeast Asia. So this is due to recurrence exposures of the typhoid causing bacteria, immunization, and high chances of cross reaction from the infections such as the malaria and the non-typhoidal salmonella. Okay, so salmonella, the other species of the salmonella is also there. So that may also cross react and give the false positive result. So if no other test, either the bacterial culture or more specific serological are available, a fourfold increase in the titer, for example, from 1:40 to 1:640 in the course of the infection, or a conversion from an IgM reaction to an IgG reaction of at least the same titer would be consistent with the typhoid infection. Okay, so this was about the typhoid and the detection of the typhoid. Okay, so the Vidal test reaction involves the use of the bacteria suspension, the Salmonella and the Salmonella A and B, treated to retain only the O and H antigens. And in this test, these antigens are employed to detect the corresponding antibodies in the serum of the patient suspected from having the typhoid fever. So the IgM somatic O antibody appears first and represents the initial serological response in acute typhoid fever, while the IgG flagella H antibody usually develops more slowly but persists for longer. So based on the application, two variants of the Vidal test are there. One is known as the slide test, and another is known as the tube test. Okay, so the slide test is rapid and is used as the screening procedure using commercially available antigens of Salmonella typhi, and a drop of suspended antigen is added to the equal amount of the previously prepared serum. An initial positive screening test requires the determination of the strength of the antibody. So this is done by adding together equal amount of the antigen suspension and serially diluted serum from the suspected patient. So the agglutinations are visualized as clumps, and weakly reactive agglutinations may require an adequate light source for proper visualization, while if strong reaction is there of the agglutination that are easily seen. Okay, so the score is the 0, 4 plus, 2 plus, 3 plus, and 4 plus. Okay, so 0 means no agglutination is there. 1 plus means 25% agglutination is there. 2 plus means 50%, and likewise. So 4 plus means the 100% agglutination is there. So the smallest quantity of serum that exhibit a 2 plus or 50% agglutination is considered the end point of the serum activity of the titer. Okay, so the tube agglutination test requires much more technical work than the rapid slide test, and it is macroscopic test. It also serves as a mean of confirming the results of the slide test. So the mixture of the suspended antigen and antibody is incubated for up to 20 hours at 37 degrees Celsius in water bath, and the agglutinations are visualized in the form of the pellets, clumps together at the bottom of the test tube, and the results are scored as in the slide test from 0 to 4 plus or 4 positive agglutinations as described in earlier. So the tube test is useful to clarify the erratic or equivocal agglutination reactions obtained by the Moore and the rapid slide test. Okay, so this was all about the Vidal test or the diagnosis of the typhoid fever. So hope you are satisfied with this detailed session. If you have any doubt, any query, you can write in the comment box. And this session is powered by Digital Version 2, Jyoti Vidyapeeth, Women's University, Jaipur. Thank you.